My name is Kimberly, and today I'll be giving a talk about the evaluation of bio-inspired ionizable lipids or lipid nanoparticle mRNA delivery. And to give a brief outline for this talk, I will begin with the background of what LNPs are, what they're composed of, and why they were initially developed, and then move into the design of our ionizable lipids, focusing on a few key characteristics that make them interesting. I'll then move into our experimentals and results, which includes the formulation conditions, how we characterized our LNPs, as well as some preliminary live cell imaging data that is very promising, and I'd love to share with you all. And finally, to end off the presentation, I will talk about a little bit of future work and directions that we hope to take. All right, so it's pretty well established that mRNA is rather difficult to deliver directly into cells, primarily because of its large size and its negative charge, which is a result of the phosphates on the sugar phosphate backbone. And these characteristics prevent it from easily diffusing into the or through the phospholipid bilayer or cell membrane and makes it difficult to enter the cell on its own. In addition, it is known to trigger the innate immune response and is rather unstable with a half-life of about just 7 hours. So, several mRNA vectors have been developed to make mRNA delivery into cells a little bit more efficient, including viral vectors, polymeric nanoparticles, dendrimers, and of course what I'll be talking about today, lipid nanoparticles. So what are LNPs? Lipid nanoparticles are a combination of a mixture of lipids and the negatively charged mRNA, which is encapsulated by said lipids, as you can see here in this diagram. And this forms a complex that has a net neutral charge, which thus allows it to bypass the phospholipid bilayer and enter cells. And this has several important applications in gene delivery, CRISPR-Cas9, and mRNA vaccines. Before I move on, I would like to take a quick moment to discuss the various roles that each of these lipids play in the LNP. So pegylated lipids are often associated with decreasing negative immune response by the body on these LNPs. Cholesterol and other helper lipids like DSBC help stabilize these LNPs. Ionizable lipids are very important as they help with endosomal escape, which I will be talking about more in depth in a future slide, so just hold on tight for that. And for phospholipids, these are what make up the majority of the LNP and fill in the gaps between the other lipids. Okay, so I briefly mentioned a few examples of applications for LNPs, but you all are probably most familiar with mRNA vaccines, especially because of the recent events with the COVID-19 pandemic. Now, the first two mRNA vaccines that were developed for COVID-19 were developed by Pfizer and Moderna and were emergency use authorized in 2020 by the FDA. But if we were to take a quick look at the ionizable lipids that they used for their formulations, we'll start to notice a few key similarities that also caught our eyes while we were trying to develop and design our own lipid library. So, first thing to note, both ALC0315 and SM102 have branched lipid tails, as highlighted here in blue. Now, what does this mean and why is this important? Well, if you are to redirect your attention to this structure here circled in red, we see a piece of mRNA encapsulated in lipids or surrounded by lipids, and this is a structure that we call an inverted micelle, which obviously has a circular arrangement. And this is very crucial if we want our LNPs to be able to effectively encapsulate mRNA and eventually deliver it into cells. But this circular structure requires the lipids to have a more cone-like shape. And this can be induced by introducing branched lipid tails into our lipids. However, as we can see with the more linear tails, we do not see this circular formation which is not as optimal for our LNPs. So that's why branched lipid tails are important. But moving back to our ionizable lipids from the COVID-19 mRNA vaccines, we also see that they have polar and ionizable heads. So why is this important? Well, the polar portion of this is important because the polar-nonpolar interactions between the lipids and the mRNA are what help keep the LNP together. So this is important for keeping it structurally intact. And for the ionizable portion, this characteristic helps with endosomal escape. Now, what is endosomal escape? It's a theory that once the LNPs have internalized themselves into the cells, it's the idea that they will, the nitrogens on these heads will protonate under acidic conditions and destabilize the LNP, thus rupturing it or disassembling the LNP to release the mRNA so it can be used in the cell. Now, this is still just a theory and has to be more extensively researched, but it's definitely worth mentioning as it's very interesting. All right, but now let's get into the drawbacks of these lipids. And the one I'm going to talk about the most here is the non-trivial synthesis, which was multi-step, spanned over several days, and did not have ideal or that great of super great yields. Now, this is not only time inefficient, but it's also resource inefficient. And if we're about to talk about this from the atom economy standpoint, using a lot of resources or starting materials or reagents to create or produce not that much of desired product, that's just not environmentally friendly, resource friendly, or cost efficient either. So what we were hoping to do was, one, keep the branched lipid tails, two, keep the polar and ionizable heads, and three, simplify the synthesis of these lipids when we were considering our own lipid library. But we also took some inspiration from nature. So how does nature make its lipids? Well, if I was to say lipid or fat, the first thing that might come to mind would be food, since we hear the words saturated and unsaturated fats all over the place, especially on like the backs of products that we buy and in ingredients lists. But what do these words actually mean? Well, as you may be familiar with already, saturated fats tend to be solid at room temperature and have no double bonds in their carbon chains, while unsaturated fats have double bonds in their carbon chains and thus have this more kinky structure that yields their liquid state of matter at room temperature. So we were hoping to include some of these characteristics in our own lipid library. But let's step away from the kitchen for a second and look at nature's other lipids. There are lots of lipids present in nature, ranging from sources like bumblebees to aquatic plants. And some examples of these lipids, like oleoyl alcohol and phytol, include, again, unsaturated portions, but also some methylated sections. So if we were to bring this all together and summarize, we, one, included our polar ionizable heads through the dimethylglycine 1-methylpiperidine course. We also included our various lipid tails, including saturated tails, unsaturated, methylated, and branched tails. And finally, as far as the synthetic route goes, we simplified it to just one step. Most of the yields were fairly decent, and this synthesis can be completed well within a day, as fast as 3 hours, which is definitely an improvement. Okay, so now that we've synthesized our lipids, we have to formulate the actual LNPs. And the method we use for this is called ethanolic injection, where we take stock solutions of each of the components of our LNP and transfer them to Eppendorfs, where we will vortex them before finally characterizing them. And for the conditions of formulations, we mostly played around with the cholesterol and ionizable lipid ratios, as based on previous literature, these were what were usually changed. So we wanted to see how different conditions would affect how well the LNPs formed and how well they were able to deliver mRNA into cells. And one of the examples of our successful trials for formulations was on the lipid dimethylglycine dioxylamine with condition 7. And as you can see here on the bright field, we see a lot of pretty nice a pretty nice amount of LNPs that formed. However, even though it's nice to see them, like it's nice to see the LNPs and get imaging and qualitative data, what's even better is quantitative data. So for that, we move to the Dynamic Light Scattering Instrument, or DLS. DLS measures the size of our particles, or their diameter, by shining a laser at the solution with our sample, and then measuring the light scattering. So at constant temperature, smaller particles tend to move faster, and thus the light scattering would have more rapid fluctuations, while larger particles would move slower, and we would see less fluctuations in light scattering. So the instrument can take all this information together and use it to determine approximately how large our particles are. And here are some of our preliminary results with conditions 1 and 4 through 12. And what we were really looking for here was to see if we could effectively reproduce the size seen from or taken from the DLS, as we see here with trials 1 to 3 in red, orange, and yellow respectively. And most of them were pretty okay, such as 1, 4, 8, 10, and 12. However, some of them did fluctuate quite a bit across the 3 trials. So what we're hoping to do is work with calibrating the DLS a little more, or we could also opt for using a standard curve with beads that have known diameter size. But yeah, that's what we're looking forward to in the future. Okay, but on top of DLS, we also measured zeta potential. Zeta potential measures the surface charge of your particle. So how the instrument measures zeta potential is by looking at how far the particles in your solution or sample travel when exposed to these 2 electrodes. So let's say you have a particle that has a surface charge that's positive. It would migrate towards the negatively charged electrode, and the instrument can use this to calculate the relative surface charge. And if we take a look at our results for conditions 6 through 9, we see that as cholesterol ratios increase, since that's what's increasing with increasing condition number, we see that the charge becomes less negative, which is interesting. Now, you may be wondering why surface charge is important. It is hypothesized that surface charge could be related to how or where your LNPs are localized, but this again has to be further studied as it's not as clearly established yet. Okay, and then the final piece of data or experiment that I'm going to be talking about is our cell imaging. And for this, we were basically trying to see how well our LNPs were able to deliver the mRNA into cells. And for this, we took some fluorescein isothiocyanate, or FITC labeled RNA, and used these for our formulations of the LNPs and exposed them to our cells and imaged them under the fluorescent microscope. And as you can see here, we see that our cells, much to our delight, did in fact have green fluorescence. Indicates that our LNPs were in fact successfully delivering the mRNA into these cells, which is very exciting. Okay, but now for future work, we want to continue running DLS, zeta potential, and microscopy experiments for the rest of our lipids and the rest of the conditions and all those combinations. And we are also looking into using flow cytometry to quantify the fluorescence and see how well LNPs are transfecting cells. And so for some more general future directions for the LNP space as a whole, I briefly mentioned it earlier, but we could possibly look into cell and organ type specificity if we were interested in directing certain therapeutics to specific areas of the body, as uh, surface charge may not be the only thing that impacts this. And next, scalability. Obviously, all of our experiments run in the lab are at much smaller scales compared to what they would have to be if we were to expand it to the market or like the public in general. And finally, storage stability is also another consideration. The COVID-19 mRNA vaccines, I believe, were stored at negative 80 degrees Celsius because they were rather unstable. So this obviously isn't very practical, especially if you're trying to transport these therapeutics to different areas of the world. So this is definitely something that could be looked into further. All right, that brings me to the end of my presentation. Really quick for acknowledgements, I would like to thank the Olive Children Foundation, ASDRP, and all of its sponsors for giving me this wonderful opportunity to explore my interests and conduct research here at lab. Thank you to all of the group mates and lab buddies I've been able to meet and work with in the past years who really make this experience like much more worthwhile and exciting, and have like pushed through the challenges with me as well. And of course, thank you to Dr. Edward Njoo for all of his guidance and really helping me through this whole experience. All right. And with that, I would like to end my presentation and open the floor for any questions.